taking form. Now I can relate it with the COVID, because everybody is bothered about COVID. People just think that COVID is as if it is the biggest enemy of the life, and the moment you have a COVID, either you have a social stigma or you are going to lose a life, which is not very true. If by social distancing, I would like to forgive you guys, forgive me please, if I'm using some loose language, but I would like to say that social distancing we talk about will help you prevent from the getting in contact with the disease. Then condom should have been the best thing to stop the HIV. But we still today get tens of thousands of new patients in India and everywhere in the world. So no matter what we say, what we do and what we say is always a different, and therefore I think COVID is one another member of the upper respiratory tract infectious agent that gets into the lung, causes pneumonia. And pneumonia is a syndrome is known to be caused by allergy, chemicals, virus, and bacteria. So it is not a one cause of it. And recently also there was one of the slide, I am just diverting myself, but I think it's important in the line of COVID. There's one video became viral from, Italy, stating that 20 autopsies revealed it is not a virus, but it is the bacterium. And a lot of people have a confusion. It is not a confusion. Basically, these are the cases which were ARDS, and the patients who were on ventilation, they basically ended their life due to secondary infection. So it is known that HIV acquires AIDS, and AIDS patient will die of anything else. So similarly, when you have a virus which has weakened the immunity so badly, and patient already had some condition due to which immunity has lost its own functionality, and as a result of which some bacterium becomes prevalent, and if you take autopsy of the patient, you would see the bacteria. It does not mean that the patient is died of the bacterium. It was started with the COVID, and COVID genomes are there, so nonetheless. But a point suffice to say that whether it is a 19th century, 18th century, or today 21st century, human is always a paranoid, hypochondriac, and gets worried about his own health. And no wonder why microbes were having a tag like the COVID today has, that it is hazardous. But today we have realized it is not true, because what microbiology we know, a small fraction of microbes are hazardous, and most of the known microbes are non-hazardous. They were treated as a seldomly believed to have a genetics. People believe that they are much of elastic in nature, flexible in nature, and that's why they don't inherit. This story was there until I think the basic definition of mutation was characterized in bacteria. And then today, if you see the basic genetics, basic molecular genetics, molecular biology, central dogma, fundamental inheritance fundamentals, recombination fundamentals have very well worked out at molecular level only in microorganisms, and no more surprise that earlier beliefs about the microbes that they are not having genetics, they cannot inherit, they are plastic, their nature is flexible, is no longer true. So what are the microbes with reference to nutrients? The majority of microbes today we are having to be cultured are unculturable. And the proportion of unculturable microorganisms account more than 99% of microbial diversity exists in the nature. Oligophilic, those require very, how many times very you want to say, a diluted environment, so that a trace of it is increased will cause a hazard, and these organisms will not go well. Auxotrophs, they are known to have a special requirement, either a hormonal, vitamin, or some growth specific, without which it can't grow in that environment, whereas a prototrophs can be cultivated in simple medium or cultivated in complex medium. So suffice enough to say that only less than 1% microbial potential has been cultured. It's not that we can culture, it's just that we haven't understood, and we haven't been able to think behind what we think presently in last 200 years. So most of the 99% or more than that, we are yet to be cultured. Amongst the cultured, only 1% of microorganisms are hazardous. which means if you take that 1% are cultured, out of that 1%, that means 1 into 10 to the power minus 4 of the known potential of the mind has its own dietary need, and the diet is there how decided. If we sway, if we keep the dietary passion at abeyance and try to adopt something else, that is what here it means by a Western diet. Don't use Western diet in later and spirit. It is just to be considered in the spirit sense. Then diverting your geographical environmental factors and towards a particular feel good, like good diet, it certainly is going to bring you into the lifestyle diseases. And this has already taken a big toll in India, particularly, which can be exemplified from the cesarean birth. Now, the cesarean birth percentage before COVID and during COVID is a very surprising. If somebody does that kind of analysis, you would see that during COVID-19 period, a large number of deliveries are reduced with the cesarean section because people could not make it to the hospital and they were delivered. Otherwise, the cesarean deliveries were more than 75%. Antibiotic use has become so common. People are looking for the options as if some antibiotic will COVID or kill COVID, or antibiotics are treated as a life-saving for anything and everything. You see a symptom, you will go to the pharmacist, and pharmacist who doesn't have a training of pharmacy but will give you the medicine and you will be taking it antibiotic. Formula feeding in an infant, it has become a fashion nowadays. But what you get from the mother's milk is different than what you get in the formula, because formula doesn't have the microbes. And the sanitation of the living environment is a huge important point. The gut microbial diversity is also decreasing. That is also attributed to the lifestyle. Because if you overwipe, if you over-clean, you are not going to have large number of organisms around you, and they have significance in training, imparting, lining your quality of immunity. And that's why the prevalence of chronic inflammatory diseases have been rising, such as inflammatory bowel syndrome, diabetes, obesity, allergies are growing. Allergy is one of the areas that we are very badly ignoring, and every day new member in allergy panel is increasing. Asthma is one of the examples of the change in the lifestyle. So what is the gut microbiome does? Then, since the immune system is developed and it is influenced by the microbial component, because if you see, if there is no microbe, there is no need of immunity in the traditional sense. But today you have a immunity for cancer, immunity for asthma, immunity for anything. So early microbial colonization is therefore a very important factor in deciding the susceptibility pattern of that individual to certain disease at a later stage of the life, as well as these evidences would indicate that gut microbiota is vertically transmitted from his or her mother so that a baby gets it. The colostrum is not rich with antibodies only, but colostrum also has certain normal flora that is a probiotic. It is given from the mother to the baby. So, however, the role of external environment in the gut microbiome and the immune development is not understood at all, or it is a very poorly understood as of now, and that is where we really have to talk about. Studies show that growing up in a microbe-rich environment, such as a traditional farm, can provide a health effects on children. I love this kind of statements in my class, that most of the people who live in a so-called sophisticated or middle class locality. Now, middle class is a fancy word used in India. There is nothing like a middle class. There's a rich or poor only. The middle class is also more or less rich people. So the people who like to mimic artificial environment, and they would like to, under the name of hygiene, remove lots of microbes from the surrounding and try to survive, automatically their immunity development is going to be compromised. On the other hand, a person who works as a maid or somebody in your house or support staff in the house who's living in the slum or living in a very, for you it might sound unhygienic environment, is basically rich in different microbes, and therefore their immune development is pretty better, and that has a healthy effects on the children. These health effects may be ablated due to changes in the human lifestyle, their diet, and a living environment, as I was saying right now. Environmental biodiversity, therefore, is a result of developing or urbanization factor. So what type of cells that we have in our body or our typical human body? So of course, our own cells, no doubt about it. And rest is microbial world. So that is a bacteria, fungi, archaebacteria, and of course the viruses. So therefore, a doctor should treat an individual as a community of organism, because although I am a human being, I have the microbes living in my gut, I have the organisms living in my every orifice, that is ear, the eyes, then the mouth, then every other organs. And what is a community of the human host through its microbes called? It is called a human microbiome. Now, where on the healthy human the microbiome is located? Every human body surface which is exposed to the environment, such as your eyes, the skin, and I think we were studying Staphylococcus aureus, Staphylococcus epididymis, and we were taught like Staphylococcus epididymis is the skin flora and Staphylococcus aureus is a pathogen. If it gets into the wound site, it causes the wound infection. And this is where you can see like two different species of a genus. One becomes a pathogen and other becomes a microflora. So similarly, eyelids have different, eyelashes have different, eyebrows have different. The eye skin and the forehead skin, underarm, the skin of your hand, the microbes can be dramatically different. Every body part on which there is an opening to the environment, such as the respiratory opening, upper airways, nasopharynx, oropharynx, then otitis, middle ear, then your eyes, then your cloacal opening, then your genital openings. Everywhere you have a microbiome. Do you think your gut microbiome would be more like a tropical forest or a desert? So it has to be a tropical forest. Normally, I ask this question in the class, and then we can go directly. Does your body contain more of your own cells or more of the microbial cell? Now, it is needless to say that 1 gram of stool contains a billion amount of 10 to the power 8 to 10 to the power 9 cells of E. coli only. So if you calculate the weight of a single E. coli and 1 gram of stool, actually the weight of E. coli exceeds the gram. And therefore it is very difficult. There is a paradoxical situation that the total amount of the stool that we excrete tells you how many E. coli that you have had yesterday in your stomach and today in your stomach. And E. coli is not the only organism that is present in the gut. So there is the issue that the number of cells in your body are definitely over in number of the microbes than that of your own body cell. So how much? At least 10 times more microbial cells are there in your body as compared to the human own body cells. And I'm not talking about the human types of the cells. Every tissue has a different type of cell, and every tissue is made up from the number of cells. Then tissues comprises the organ, and organ together multi-organ becomes an organism as a human. So even then, the number of cells the human has and the number of microbes in that human, on that human has, the number of microbes is always at least 10 times more than the number of cells of the human per se. The combined weight of all microorganisms in the body can weigh as much as very several pounds. And the best example I can give you, somebody is sick, hospitalized, and treated with certain antibiotics. So basically, it is very clearly known that antibiotics do not affect the eukaryotic cells because they are targeted for the prokaryotic, and in particular, the cell wall of the bacterium, or commonly used beta-lactam cell wall, and some are targeting the 70S ribosome subunits or the certain membrane component of the bacterium which are missing in the eukaryotic membrane, eukaryotic translational machinery is different, or eukaryotes, the human doesn't have the cell wall. So therefore, you can think that these antibiotics should not reduce the weight of a person, but you would see that invariably most of the people who are hospitalized and administered with the antibiotics for a couple of days loses a weight by several kilograms, and that is basically losing the microbial flora, and that results into the reduction, significant reduction into the microbial count, which reduces the weight of the person. And after restoring the normalcy of the life and regaining the capacity of digestion and metabolism, the person regains the weight as it was before the hospitalization. This is a very clear indication that indeed the microbes account for our several pounds or several kilograms of our mass. What are your microbiome organisms then doing in the body? They are living in a complex ecosystem where microbes can play different ecological role, such as large number of unknown functions for which you have to indulge doing research or something and try to understand what they are doing. Yesterday, I was talking to one of my classmates, PhD classmates in Ohio, and he was telling that when he realized that I was working on human microbiome, then he said, our lab is working on lung cancer, and then they have found a cell of a cancerous person, in the particularly the malignant part, that they found a bacterium present in the cancerous cell, and that bacterium makes person respond positively to the anti-cancerous molecule. So here is a very good example that they still don't know what that bacterium is, but a bacterium is able to reach to the lung, which is not a normal thing, because normally lungs should not have the microorganisms. But if a bacterium enters in a cancerous mass, it is basically helping that human to absorb the anti-cancerous compound and kill the cancerous cell. So basically it is making individual respond positively to the anti-cancerous molecule. So similarly, there was another interesting paper that I read, Sapna brought, then a point mutations in E. coli of an individual basically imparts the colon cancer. So already Helicobacter pylori was triggered. Now, if they had 10 patients identified with that cancer, and then they characterized the E. coli genomes, and then they found that all of them had that particular mutation. They even augmented that mutation into the normal E. coli, and in mouse they studied that if you impart this mutation, yes, indeed, it can trigger into the cancer. So you can see that changing the normal microbial genetic composition is not going to be healthy for you. So therefore, what is more important is that nature given microbial composition has to be maintained. Today, there are large number of papers in last 15 years talking that microbiome is linked to the secretion of large number of pheromone, hormone, and thereby the behavior, psych, brain functioning, in addition to actually maintaining the metabolism, then oral microbiome, and then overall your health. And therefore, I think it is a lot to discover organism-wise, and then the diversity at a genetic level of those organisms doing what kind of functions. Then predator-prey activities are going on in your body, because every cell is regenerated, except the neurological cell I'm talking about. They are died, destroyed, new cells are born, then come to the maturity, then they are dying, there is a senescence, and then those dead cells are being taken care of. So kill our own cells or our other microbial organism, that is a predation and prey. It is to become a prey to the other organism so that it would be eaten by the other microbes. And those microbes may be required for the predation. So therefore, becoming prey is also the role. Now, certain microbes are known to be a producer. They make certain vitamins, certain chemicals, and which is required for the growth of our other microbes, or it is required for signaling of the human various activities. Decomposition, as I said, the food and waste that we generate in our body, basically decomposition is required, and that is the biggest primary role of human microbiome. पुराने जमाने में कहते थे कि अगर ससुराल जा रहे हो तो पति के पेट का ठीक से ख्याल रखो, पति का पेट अगर ठीक से रहेगा तो उसकी साइकोलॉजी ठीक से रहेगी, उसका काम में मन लगेगा और ऑटोमेटिकली सब चीजें नॉर्मल रहेगी. How well, how carefully it has been mentioned that the gut microbiome is important for your whole behavior, for whole health and your thinking also. So now here is a lower and higher oxygen content example. Now lower oxygen content basically results in the increasing the H2S production. So plant fiber or animal tissues are being eaten up, degraded by the primary degrading bacteria. They produce certain partial degradation, produce certain other carbon sources, which would be used by the secondary degrading bacteria, and then those will again make substrate for the variety of other bacteria that results in the production of small chain fatty acids, methane, acetate, hydrogen sulfide. But when you have a lower amount of oxygen, it would result in higher amount of H2S production. Now, higher oxygen basically results in reduced amount of H2S production. So basically, the host microbial gene expression is so important. So it's not just a host microbe interaction, host pathogen interaction, but even a host normal flora interaction and their gene expression, if it is in a cordial manner, then the health of both microbiome and in turn the human can be guaranteed. Now, what is the gut microbiome to do with the immunity? Interaction of microbes in the gut, that is intestine, early in the life will devise or train the immune system to distinguish self and non-self. So there are some organisms, they are only meant for discriminating the group members and non-group members. Presently, autoimmune is one of the biggest things that we talk about. There is a large amount of blindness being developed in the body, and that's why the self is being regarded as a non-self and causing a large number of autoimmune diseases. But that is also because some of the organisms might be in a too small concentrations, are meant to be there in gut microbiome, but they have been extincted or diminished, and as a result of which the autoimmune is triggered. So microbes and the medicine, let's say throughout your life, microbes are known to secrete the compound that regulate our immune cell, particularly T cell, such as cytokines, chemokines, and certain other chemicals. Now, microbiota has been studied. A conceptual of commensal organism was very old in 19th century, and recently, as I was telling you in last 10-15 years, there are a large number of human microbiome papers. 1 to 3% of the total body mass is accounted by that, generally non-pathogenic, and they are symbiotic with the host. Now, this is one of the papers from the class 1 telling that the relative proportion of bacteria bacterial produced short-chain fatty acid differs significantly between the stool of a healthy person and individual with the colorectal cancer. So there is like a light color and a gray color, basically the colorectal cancer person. You can see acetic acid is increased, then propionic acid is to some extent increased. There is a decrease in the butyric acid, valeric acid is increased, isovaleric acid is also increased. So these are the reasons. So if you know which organisms are required to bring down the acetic acid, valeric acid, isovaleric acid, propionic acid, and trigger the secretion of butyric acid, then the transplantation can be worked out. I am not talking something that you should be unaware of. There is something called a fecal transplantation. Here there is a hospital in Delhi called ILBS, I think Indian Liver Biliary System Hospital. There's a Professor Sareen who has performed few tens of fecal transplantation. Don't get nauseated with the word fecal. Fecal transplantation, basically here it means that healthy human microbiome is transplanted into the patient. He tried onto the irritating bowel syndrome and he has found it very useful in the therapy. I think this is what is very important. So if you know that which organisms will restore the acid balance, then balance of the acid will trigger or force the person's colorectal cancer condition to reverse, and if it is required, one can have this transplantation carried out at different time point. It's not a surgery, it is basically the administration. Natural bystanders, therefore, are so important. Natural bystanders means what? Your normal flora. They are known to guard you against pathogens. And this is very well known. Your nose is not sterile. Your oropharynx is not sterile. Your ear is not sterile. It has its own flora. Your skin is not sterile. But too much of body shop, too much of perfume, too much of creams basically is going to remove your normal flora. I know one of my landlord's mother, who is probably in her late 90s. She has never used soap in her whole life, but she never smells any kind of bad. She was one of the most healthy person I have known. So you can think about it. Your body knows how to take care of. And we have given all that contract to the body shop products, and it has caused what? If you don't use them for 2 days, you're going to have an off smell. That's because the bad guys will prevail the good guys. So bacteria guards against HIV was published in 1994 in Science News. So it's not like tuberculosis vaccine can protect against COVID. HIV, anti-HIV molecule can protect somebody from COVID. So there is a cross-reactivity, and bacteria which are normal flora, if you have, then it will also make sure that the HIV penetrance will be reduced. Very interesting. Now, what is more you have? Obviously, as I said, you have 10 times at least microbial cell, the microbial genes and microbiome genes have to be much more in number. Now, who's in control? There are 100 times of more microbial genes present in our body as a microbiome than our original genome, that is 3 into 10 to the power 9. Microbial genes are known to turn on and turn off the response to what we do. Just like a lac operon, you have a lactose in the environment, lac operon is turned on. The glucose is there, lac operon is turned off. Similarly, these microbiome, they sense the sensor molecules in the different physiology, and accordingly they will synthesize some molecules, which will basically help trigger the endocrinological secretion. This is now a big area people are working on, so psychology and endocrinology, that microbes in the human body can decide on which hormones, which pheromones are to be synthesized and which pheromones are not to be synthesized. Our genes, let's turn on and turn off in response to what our microbes are doing in our body or on our body, and which is happening without our knowledge, that is involuntarily happening. What information would scientists then use it from? A list of names of gut microbes present, a list of major enzymes which are operating in the gut microbiome, that we can make it out. Then what do you do that might change your microbial community? That's very important. As I already said, a fecal transplantation or some other sort of things. Now, our microbiome is not known to be stable. It is meant to be more flexible, more modulable, and as we grow with the age as well as the maturity, the microbiome quality changes. The puberty and during pregnancy also the quality of microbiome is changing. One of Sapna also told me a few days before that, just before the COVID lockdown, that the thing that we always thought that bacteria cannot cross the placenta. This is very well said statement in microbial things. And even except IgG, other antibodies don't cross the placenta. This is what we teach to the student. But then she asked me whether before birth there is a microbiome, and I clearly said no, Sapna, there's no such thing. And then she said that there was a paper wherein they found that there are 2 or 3 microorganisms that are exclusively meant to be there as a microbiome of amniotic fluid. So you can imagine now people are going very crazy and trying to indulge into very interesting endeavors and trying to come up with a very fantastic outcome. So our diet change will also force microbiome quality to change. Medical conditions and the treatment for the medical condition will decide. So primarily your lifestyle decides what medical conditions you will have. The treatment will then decide what medical microbiome quality. So because it's a very flexible. Pets in your home, because when you are growing, like I was telling that microbe-rich environment, if you are growing, then it will be good for the children to develop the immunity. Similarly, if you are living in the house with many pets, of course, your body will try to acquire some of the microbes, which will make you survive and make you comfortable with these pets in the house. Many more factors are being investigated. So microbiome changes, basically kinds of microbes present in the environment, can be considered as a qualitative change, number of each of the microbe, as well as a relative amount of each microbe, stoichiometry. So there is a proportion, like there are like thousands of microorganisms present. Out of that, few tens can be cultivated and rest of them cannot be cultivated. But their proportion is fixed. And if the proportion is disbalanced, which will result in some time one or other kind of medical condition. So it's not just that they have to be there, but they have to be there in a relative proportion. If the relative proportion is being disproportionately changed, then it will also result into the physiological disturbance of a human. So kind of active microbial genes is also very important. Compare your most recent meal to the classmate's most recent meal, and you can see how change you are likely to get it. Predict how your microbiome may be different as a result of you take something different in your meal. Now, what are the methods used to do this? Many of the our fellow traveler microbes are yet to be identified. That's what I told you. In the past, to study the microbiome, scientists had to grow them in the lab. They would identify and characterize the bacteria by their colony characters, biochemical characters, etc. But I already told you in the beginning, we have not been able to cultivate more than 99%. So this approach is definitely going to be a very limiting, will not suffice, and will not ever result to the fullest of the possible. So definitely one had to identify the best approaches. Microbiologists also identified the microbes by their physiological characters, such as levels of oxidation, different staining methods, cultural methods also. Now, what are these organisms which have never been grown in the lab? So in the 1980s, the scientists developed ways to analyze the sequence from the microbial DNA directly, what is called a metagenome or microbiome. So it is impractical to fully sequence every microbe in the human microbiome, because if you think that there are 2,000 to 3,000 genes in a microbe we have, and if you have thousands of microbes, basically we are going to sequence billions of the genes for no reason, just to identify. So that is not practically possible. And even if you do that, what would you do with the other extra information? So you just have to know which organism it is there. So you don't have to know the genome of those organisms. So therefore, an alternative molecular mechanism had to be used. So DNA sequences can be used as a marker to categorize organisms into taxonomic groups or broadest narrows. So domain, kingdom, phyla, class, order, family, genus, and species. Thus, the two organisms can be different from one domain of the organism to the other domain, which can be discriminated on the relatedness in the DNA sequence. And the more related DNA sequence can be grouped into one, and the dissimilar can be grouped into the other. So what are the methods available for that? Ribosomal DNA sequencing. You can sequence the RNA polymerase, you can sequence the elongation factor of translation. And for our purpose of the human microbiome, I think 16S rRNA is sufficient enough. We don't have to go for elongation factor or RNA polymerases also. So what does RNA 16S means? Basically, it is the related to the density of the Svedberg's unit and density of the sedimentation. So certain sections of the 16S RNA have same DNA sequence for all the known bacteria, which is most conserved region, and other sections have the variations, hypervariable regions. So this is taken into consideration. And if we have a base change in the conserved domain, which will tell you the evolutionary background. So there are two approaches. I would like you to see on the left side of the figure, in the central part, microbial community and sample. There are two arrows which goes down and up. Both of them are basically for isolating the chromosomal DNA. One talks about a metagenomic approach of the shotgun, and other talks about the 16S approach. So what is a shotgun? Basically sequencing the whole genome in a different length, and then aligning all those lengths so to get the whole genomes. But as I already told, sequencing the whole genome will again be used just to identify the name of the organism. And rest of the sequences will be unnecessarily, we have spent time and energy and money for doing nothing, for gaining nothing. On the other hand, on the top panel, that is 16S based sequencing, you can see amplify the 16S sequence that is shown into the two lined ones, colored ones, and those sequences are then checked into the database, and you will be able to differentiate what are the organisms. So basically, single nucleotide polymorphic sequences will be able to tell you which organisms are there. So whether you go for the whole genome metagenomic approach or 16S metagenomic approach, you will end up in identifying the organisms. So more importantly, if you see into the graph that shows the abundance is shown onto the y-axis, and OTU is shown onto the x-axis. So you can see that units, that is units can be seen that the red one is highly abundant, then comes the blue, the orange comes the third, and rest of them. So here abundance in a certain standard organism, you would be able to, or a standard person, you will be able to relate it as the normal one. What we are doing in Ayush project is basically we are taking 180 patients and healthy people, and their abundance will be average wise calculated as a normal, more or less, the normal microbiome for the healthy human entity. So what might be some goals for the human microbiome project? Basically, develop a reference set of sequence. That's what I said that we are doing it with the Ayush Ministry. And a preliminary characterization of the human microbiome. So here we are taking the 180 people who are going to be collected specimens in Uttarayan and Dakshinayan. They are between the 18 to 40 age. They don't have any kind of genetic disease. They don't have a history of genetic disease. Their do's and don'ts have to follow, their diet, etc. will have to follow. So 180 getting sample in itself will be a big challenge for us. And their human microbiome will be considered as a reference sequence for the human genome of the Indian subset. So provide information about disease and microbiome changes. So later on, we can do the same exercise on oral cancer, we can do the same exercise on lung cancer, we can do the same exercise on different lifestyle, obesity, asthma, and then try and see what is the disbalance in the proportion. Or is there something is missing from the normal and something is present into the patient? So that's what we will be able to find out the solution. So develop new technologies and tools for the computational analysis of this high big data. Establish a data analysis and coordinating centers. Establish a research repositories for all the sequences and all the organisms. Examine the ethical, legal, and social implications of the human microbiome project research. Evaluate the multi-omic data to understand the human microbiome's role in the human health and disease. Multi-omic means here it is basically proteomic, transcriptomic, metabolomic, along with the microbiome. So we will take all these things together. So systematically, system analysis will be doing it. So who all are involved in microbiome? Not just microbiologists, an immunologist, a microbiologist, a molecular biologist, an enzymologist, a taxonomist, an ecologist, a computer scientist, a biochemist, a physician, a mathematician, statistician, a social scientist, and of course, a ethicist is also required because everything has to be ethically right. So to recap, human microbiome is of a community of varied microbes on their human host. Microbes can be 10 times more than the human cells. There are hundreds times more microbial genes present in the human body, which is known to be regulating the immune system health and the behavior of humans. Scientists have cultured the microbes to isolate the organisms, DNA, and then 16S sequencing to analyze the human microbiome. Hundreds of different scientists, mathematicians, computers, social scientists are working together to give the expected outcome of the human microbiome project and to develop the methods, tools to understand how this fascinating topic is. In addition to this, I would also like to bring it to your kind attention that microbiome concept is not merely the human microbiome, but it is also relating to your plants, what we call like endophyte, then mycorrhiza, not mycorrhiza, because mycorrhiza talks about fungal, but mycorrhiza will be the organisms which are present in the surroundings and how do they basically help the crop or the plant to grow. So we can think about microbiome in animals, microbiome in plants, microbiome in environment, and I think possibly we'll be able to talk about soil fertility, agricultural aspect. We'll be talking about environmental quality and organic farming and a better suited environment in coming future. With this, I would take if any questions are there. Thank you, Darshana ma'am. Thank you, Shubha ma'am. Thank you, Rashna ma'am.
Thank you, sir. Hello.
Yeah.
Yeah, thank you, sir. Sir, I have 4 questions.
Yeah.
Yeah. So the first question is, how about FMT working in India? So FMT, that is fecal-medical transplant. So what the way you said, so it's very, this concept is very common in USA and they are going ahead. But since we don't have a reference of organisms, like what organisms are ideal for our gut, so how can we suggest FMT? That is the first question. Okay, because considering what research you are doing at 180 people and you are doing, so we don't have a clean sheet which says that this organism has to be there in the ideal gut microbiota. Okay, so how come we can take, how can this transplantation be done and what will be the side effects of it? The second question is, if a lady is undergoing a cesarean delivery, okay, so is there a practice doing where her vaginal swabs are being or her vaginal washings are being absorbed on the baby's skin or not? Because what I heard from there was one talk where I heard that this thing has to be done wherein if the baby is not coming through normal path, so he is not acquiring the normal flora or the all the vaginal organisms. So for that reason, there are some studies also going on where they take this swab and they just allow it on the skin of the baby to absorb if it is cesarean section delivery. The third question is, sir, how non-veg eating will affect this gut? Yeah, will it improve or will it affect it badly? Because I don't know. In both the cases, I don't know. And the fourth question is, sir, what is your what is your opinion about how to handle this project? It's such a big project. So how can at college level students, by doing their MSc project, how can they contribute towards this topic? Because, sir, it is very big. So it can our college students, by doing a big part of it, and they can connect with you. So how can this social outreach of your institute of from your side can help our students to reach out to you as well as do a part of it? So these are my 4 questions, sir. Thank you.
So the very first was a fecal transplantation you were asking. You rightly said that we don't have the reference organism list, not just the list, even the consortium. I think list is not so important. List is not going to be big difference in a person with a condition as opposed to the person who's categorized as healthy. What is important is the proportion, that is the stoichiometric proportion. So at this moment, what ILBS does is they have people who have been identified not to have any kind of conditions, and with all the possible testings, they are considered to be the healthy. Their the purification of the organisms by the microbiologists is being carried out, and that is being used as a fecal transplantation. One has to wait till we make a reference line on the data as of now, and I don't think we have any such kind of data in India. The Indian data is available on the genomic level. So there was a project from IGIB New Delhi. They have carried out the Indian genomic studies, and that data will tell you whether the human genome, which we know already, and the Indian genome matches or not. So that in itself is giving a lot of hurdles to people, and but once we have that clear-cut understanding, they will see how different our genome is. That's answering to your first question. Second was a cesarean birth, and then let's say brushing or swabs to be given. It's you can say theoretical is there, but even in USA that is not practiced. It has to be under a controlled trial. It's not a general practice method. One. Second thing is that when baby is coming through the birth channel, it has its own path and the interaction with the flora. So the way it will acquire the flora, adhere to the scheme, is certainly not going to be the matching with the, no matter how much of the vaginal lavages you take it for creating a sufficient coating onto the baby. So it can help, but it will not exactly mimic the situation of the natural birth. Natural birth is natural birth, and that's even there also you see some babies are born. Transition phase is very important in delivery. Transition phase normally lasts for 30 minutes to 2 hours from one patient to the other patient and from each delivery in a person also. And if some babies are born in 10 minutes, though we say 30 minutes to 2 hours, and some babies are in transition for like several hours. So there, is it absorbing of the organisms and exhausting the organisms from the path? It's not like that. So just getting into the contact and the path in itself, the all organisms, how sure you are that your lavage will assure that the all normal flora of that birth canal is being transferred into the lavage. So there is a lot of questions that are unanswered, and that's why it is not a general practice. But yes, a naturally born baby starts to acquire the organisms during the birth itself from the birth canal, and then the second from the colostrum. That's true. And third, obviously, wherever the baby is kept and the environment also adds into the organisms onto the scheme. What was your third question? Was a diet?
Yes, sir. How non-veg diet? Yeah.
So I think I have already discussed the point that diet is evolved based on your geography and environmental situation. So we should not be categorizing it as a vegetarian or non-vegetarian. So what is more important is that what is the nutritional requirement of an individual where you reside. So depending on the situation out there, your requirements are going to be there. So particularly in India, where you can see the coastal people won't get as much vegetable as much we get it in, let's say, Satara or other parts of Maharashtra. But you really have to think about what would they get, and that's how they have to adapt, and their body is also adapted. It's not just because the vegetables are less, so fishes are more, so they are eating fishes. So no, it's also the environment out there, the humidity, the salinity, the other chemicals, physical parameters which are present in the environment. Those are also important on your overall behavior and the physiology. So I think we should not categorize as to the vegetarian and non-vegetarian. Coming to the last and very essential important question. How do we connect our undergrads with the microbiome directly? I think it's a very wise question, smart question also. Because the technology is such that it requires not much of the things. Even I don't do it in jail. I collect the sample, I do it for sequencing, then they give me the data. So what is my role? My role is actually the analysis of the data. So one has to be good at the analysis of the data. Because you cannot have the sequencers given in every university also. How many universities of Maharashtra has a sequencer? And if they have a sequencer, how efficiently they are doing? NGS, particularly I'm talking about. It's not a practical solution. But what is more important is that starting from Sangor reaction till the NGS, if my student is very well versed about the technology, then any sequence information he or she is going to get will be in a position to analyze. So it is very important at this level for us to train them very well the basic principles of sequencing till the advanced sequencing methodology and analysis of a big data of sequence analysis. So that can be done in silico in the absence of the such a big fascinating technology. Also, these students can, based on the scholastic possibilities, can work with those institutions or those companies and try to have the hands-on or at least view the experience, how these machines work, how do samples are being processed, because mostly what is crucial is like sample preparation is very crucial, because a very few people can handle the instrument. Okay, surgeon can deliver the baby by cesarean, but not every doctor is efficient enough to deliver naturally. But a nurse can, because she has a profound experience and she has a technicality. So similarly, how you make a sample, how you pay attention while making sample, and that's something a student can be trained in the institutes. They can be taken up these kinds of what is called like internship, and they can learn about. But it's a growing technology, and basically India is lagging behind for 2 decades at least.
Yeah, thank you, sir.
Yeah, thank you very much.
Sandesh sir, you can take the question. Sandesh sir. Sandesh sir.
Ma'am want to ask.
Really speaking, there are no questions from the students as such. I think everything has been well explained.
Sandesh sir, there is one question.
Yeah.
Sandesh sir, there is one question from Kumar in the chat box.
One question is there, sir, from Sneha Kumar. So, sir, we should reduce the use of chemical products like cosmetics, artificial environment.
So there is echo voice. Sandesh sir, there is echo voice in your. We can hear your echo voice.
Are you able to hear me, sir?
Yeah, now I am able to hear, but there is some disturbance. Yeah.
Okay. The question is like this: should we reduce the use of chemical products like cosmetics and artificial environment? To what extent?
Oh, that's a very wonderful question, actually. But very interesting thing is that we cannot start practices as and when we like it. That has to be monitored, mentioned, because as I told about developing the microbiome, it is beginning from the time of your birth, and then the early few days, and then sets up. Then it changes at the puberty, then it changes at the changes in the physiology is there. So actually you can't give up the chemicals directly if you're so used to. You have to try and reduce them slowly, gradually, and then maybe you will come up to a point where you can survive, or you can completely get out of those chemicals. But yeah, try to reduce them as maximum as possible, and probably you will be coming to the only essential things, because your body is born with all the mechanisms. Your body is born with all the mechanisms. Try to live in an AC-free environment. Initially you'll have a suffering, but after 2 months, you will realize that, yeah, your body knows how to make it. So, but now lots of mutations have been topped up in individual, in me, in you, and everybody. So probably everybody is not though we are theoretically expecting, practically it is not likely that everybody will cope up with. So one can try to give up the chemicals, but see to that you don't end up into the problem. So you have to monitor, but not all of a sudden. You have to gradually reduce them.
Okay, that is all. No more questions, sir.
Okay. Thank you so much, Dr. Kharat sir, for guiding us in this very huge topic, and it's a ever going topic. And since you said that we are 2 decades away from India is 2 decades away, but sure we have a belief in you all scientists as well as teachers and our students that we will surely come up with a we'll speed up and the pace of the data very soon, and we'll give all help as well as we'll take all the knowledge from you as well when the sub project is done. Thank you, sir, for such a good guidance as well as help towards enhancing a lifestyle as well. Sir, I just request Rakhi ma'am to go ahead with the next session. Thank you, sir, and thank you, ma'am.
Thank you, Darshana. Good afternoon, all. I, Mrs. Rakhi Saudankar, I'm glad to welcome today's second speaker, Mrs. Sonia Nandre Patil. She is the General Manager, Laboratory Operations, TUV India Private Limited, which is a member of TUV Nord Group, Germany, a leading technical inspection, certification, and testing organization. She has more than 20 years of professional experience in the field of laboratory quality assurance and laboratory operations and management. Worked at NABL and also coordinated in food, pharma, and clinical laboratories accreditation. NABL in panel lead assessor and technical assessor for biological discipline. Conducted more than 80 audits as per ISO 17025-2005 on behalf of NABL. She has also done the documentation, implementation of ISO 17025 quality management systems for testing laboratories. It gives me immense pleasure to introduce her briefly. She is MSc Microbiology, pursuing her PhD from Pune University. In the journey from the Junior Production Officer at LG Industries Pune to the General Manager TUV India Private Limited, there were many milestones. She worked as a Research Associate, Indian Agriculture Research Institute, New Delhi, Junior Scientist, National Institute of Biologicals, New Delhi. She worked as an accreditation officer with NABL New Delhi, quality manager at National Agriculture and Food Analysis Research Institute, Pune. She was assistant quality manager with Govlikar Metropolis Health Services India Private Limited, Pune. Also as a manager QA in a National Agriculture and Food Analysis and Research Institute, Pune. She joined TUV India Private Limited organization in June 2009 as an in-charge laboratory operations and progressed from in-charge to head QA to manager of laboratory operations to deputy general manager of lab operations, and currently working in the capacity of general manager laboratory operations in the TUV India Private Limited. During her tenure in TUV, she was involved in the various activities. She worked as a key member in overall planning of the design of laboratory, identifying the equipment with specifications. She was involved in validation of test reports, interaction with all government agencies like NABL, AGMARC, EIC, APEDA, FSSAI for their recognitions. Evaluation of validation procedures and the data of compliance for the methods like pesticide residues, toxins, and other food parameters on GC MSMS and LC MSMS. She has participated in various trainings, workshops, and seminars. To name the few, she has participated in training course for training the trainers for GLP compliance organized by FDP, UNDP, WHO at New Delhi, lead assessor course in ISO IEC Guide 25 at SGPGI Lucknow by IIQM Jaipur and NABL. She participated in a course in laboratory QC/QA and biosafety program by TCMR USA at National Institute of Biological New Delhi. Workshop on the College of American Pathologists accreditation at SRL Ranbaxy, Mumbai. Workshop on quality assurance networking for HIV, on quality assurance networking for HIV testing laboratories in India at National Institute of Communicable Diseases, New Delhi. We all are very glad to have you here today, ma'am. And I know that all the students and the participants are eager to hear you. So I kindly welcome you and ask you to begin the talk. Thank you, ma'am.
Yeah. Thanks, Ms. Rakhi, for that long, elaborate introduction of mine. Not much needed, actually. It could have been shorter, but that's fine. One question, can everybody see me?
Yes, ma'am.
You can see
We
me?
can see
Okay, fine.
the screen.
Because my camera was not on, I was unable to check whether I was visible or not. So good morning, everybody. Rather, it's good afternoon right now. So I'll just share my screen. I'm going to introduce the opportunities for microbiologists. I'll stick to that topic. I don't know the students' background, but I'm considering they'll be of microbiology and allied. Some people are saying I'm not visible.
Yeah, ma'am, you're not visible.
Yes, you are not visible.
So now, because the camera control, it's not in my hands. Anybody from IT can do that?
Is your camera on?
Yes, I have. Just give me a moment. Is it visible right now? No.
No.
No. Just give me a minute. I'm just trying. Can I rejoin and just see whether I can do it?
Yeah, please try that.
Yeah, I'll just try. I'm sorry. I don't know the camera is
No
not on.
problem.
Just give
No
me a
problem.
moment.
No problem. Take your time.
Am I visible now?
No, ma'am.
No, ma'am.
I think I'll go ahead now. Because I tried relogging it, I'll just go ahead now.
Okay, sure. I think you can just start your presentation and
Yeah, sure.
Yeah.
I'm just sharing my screen a moment.
Yeah, please.
गूगल नहीं है एक सेकंड उगड़ते
मुझे ड्राई रन कर देना।
नहीं कर रहे थे
Enter screen.
आईपैड वर घेतलीस काय
तू कर प्रयत्न, मी तुला फॉरवर्ड करते. त्याच्या विनीत का आलाय बघ. Dr. Rajeshri, I am not able to. Can I speak then? I don't know. I'll try. I'm trying it on my iPad as well.
Ma'am, you can send your presentation to Vineet.
Okay, fine.
So he can
Is it
display-
visible right now?
No, ma'am, not yet. Ma'am, you can send your presentation to Vineet so he can display it
Fine,
on he can share the screen and you can talk.
Yeah, sure. In a minute, I'll do it. No problem.
No problem.
I'm very sorry about this.
No
We have
problem.
done a dry run with you yesterday evening. I'm so sorry.
Ma'am, that's a technical problem that happens, ma'am. No problem. No issues.
आणि याच्यावर तुझं हे नाही काय करायचं
Vicky, did you get my presentation?
Yes, ma'am, he got it.
Yeah. So, good morning, everybody. Till the screen comes on, I'll just brief you that today we are going to look the opportunities for microbiologists in the food industry. And I don't know the forum here, there must be some chemists, but the food industry is equally good for people into chemistry, biotechnology, and other allied life sciences, nutrition maybe for that matter. So everything related to life science and food goes hand in hand. And food industry is an extremely upcoming industry which has no recession. The demand for food will never end. So my first slide, I don't know, it's still coming on the screen, says this is the sunrise industry. And yes, because it will keep on growing. So just I'm waiting for the slides to come up. Yeah, Vineet, can we go to the next?
Hello, ma'am. Sonia, ma'am. Hello, Sonia, ma'am. Are you there? Sonia, ma'am. Sonia, ma'am.
She's not audible. She's there in the meeting, but she's not audible.
Yes, ma'am. She's not audible. I'm trying to.
Rashmi, can you please call her and ask her to rejoin? Rajeshri? Rashmi, madam?
Ma'am, yeah, I think ma'am is not able to speak or switch on the speaker. I think she will call if you have said her. She will call.
Okay. So we'll wait, है ना? Okay.
Am I audible now?
Yes,
Am I
ma'am, you're
audible
audible.
now?
Yes, ma'am,
Yes,
you're
ma'am.
audible.
You are audible.
Please go to
Yeah,
the next slide.
Please go
My
ahead.
request, yeah, the next slide. I'm so sorry, you people are being very patient with me. I'm extremely sorry for this.
Not
We'll
a
go on to
problem.
the next slide. Go on to the next slide.
Uh, who is controlling? Vinit, are you controlling the PPT?
Vinit, sir.
Vinit, go to the next slide, please, Vinit.
I just call him. Okay.
Hello, Vinit, are you there? Hello, Vinit.
Even his speaker is not working. We'll just yeah, he changed it.
He has changed the slide, huh?
Yeah, but he has gone on the fourth slide, but I want the fourth slide. He's gone a bit too ahead. Can we go back, Vineet? Another 2 slides behind. Yeah. One more. Yes, fine. That's fine. So we have said the food industry is the fastest growing industry. Food processing has become very, very important nowadays because of the urban culture coming in, women working, traveling. So processed food industry is really gearing up. In 2016, it was 332 billion industries and expected to grow nearly twice, that is 543 billion in 2020. So just imagine 1 billion is equal to 100 crores. So imagine it will be several thousand and lakh crore, and it is the fastest growing and will keep on growing. So the bar and the arrow which we have shown will always be in a growth stage and not even stationary. So there are some businesses which can be stationary after growing sometime, but food will never be stationary. It will be fastest. Move on to the next, Vineet. Yeah. So food industry is in India integral. I can say farming is an integral part of Indian profession, which is not given much importance because of the rural area, and there is no like professionalism or organized sector in our farming area. Unlike other businesses, maybe like the industries, auto industries, maybe other IT industry, may be very, very organized, but food sector is not that very well organized. The turnover can be much more, but we are not able to quantify because of the unorganization in the industry. We need next slide, please. Can we go to the next slide? Yeah. So the salient features, we have missed one more behind. We have missed a slide. Yeah. Processing industry, we are the 6th largest food and grocery market. One is because of the population and the other is because of the land diversity, eating habits, everything. So India is the 6th largest food for grocery and market. 65% of our total food is attributed to food and grocery. So everything is coming in 6th largest. 65% of our total market is attributed to food grocery. Means what? If there is an auto industry, if there's a market for clothes, there's a market for auto industries, there's education, everything, there is a market. But out of 100% of total India's, there is travel industry. So you can just imagine, out of 100, 65% of our total India's market is for food and grocery. So there are 65% jobs also increase, like we feel opportunities, okay, in any sector. So out of that, 10.7% of exports in India from food and agriculture. So India exports huge different types of materials abroad, right? From you get small units of manufacturing biopesticides, maybe fertilizers. The government has excellent schemes where you can really find out that they are promoting entrepreneurs for little processing units, biopesticides, fertilizers. So can be one of the entrepreneurs also to begin with. Then there's waste management. So a lot of waste is generated in food industry, as well as even every household generates so much of organic waste. So waste management is important since it's on organic waste. Microbiology plays a very important role. So microbiologists also can have a role in waste management of organic produce, right from the industry, from household to above, like vermicomposting, maybe biodegradation. So most of the plants have a lot of lignin. There's so much of research going on lignin degradation. These are a lot of environmental sectors where even the pure science people, life science people can take part. Last one I have said is sustainability. Sustainability is also being promoted a lot by the government. What is sustainability? Like reduction in plastic, use of paper, biodegradable material for little packaging, as well as even throwaway. We are trying to promote sustainable products like leaves or maybe little paper cups. So paper cups, as I said, seem to be biodegradable, but from inside there is definitely a lining of polystyrene, polyethylene, or any of the plastic material. So those who claim that this is a biodegradable product, who really knows whether it is or not? So even to do this biodegradation of the packaging material, there are a lot of methods, lot of research is going worldwide on degradation of really how biodegradability is there, what happens to a paper, what happens to a paper cup. There are a lot of standard methods also to check on the degradability of claimed biodegradable products. These are all emerging new fields coming into. So I'm going to the next, Vineet. So how will you prepare yourself or equip yourself to face the industry or face what is after you finish your graduation? Few of you must have, but these are few of the things which I have learned when young students come to our organization or when I go and meet people. So first of all, make goals. I don't say make goals for 10 years or 15 years, no need. Make goals, little small goals also. Maybe you may have your big dreams. Of course, you need to have, but we have to reach those dreams through little small goals. So first, in knowledge and education is very important, which you're already getting in your curriculum. But trying to get internships, try to face the industry. It doesn't matter whether you're being paid or not, but please, I feel that you should go and look at the industry, work for the industry. See, when you go and work in the industry, what type of experience you get. Nobody will, even if you pay any amount, nobody will give you that type of training or that type of experience. So I encourage students to go do internships in the industries. You also should have self-development plans like improve on your competencies, skills, and abilities. 3 things, very, very important. So first is improve on your competencies. Yes, competencies can be anything, good learnings, fast learnings, grasping power, maybe skill, competency in subject line. If I say food safety, develop in food safety. If you say spoilage, develop competencies in spoilage. Go read through, see what are the latest trends in the industry. So develop your competencies in whatever you want to do. Next is skills. Skill is an art. It's what most of the, I would say still 60% is what you have inborn and what 40% you have to develop. So I usually give an example. Suppose I like to do hand embroidery and I keep on working on it and I have no skills for that. So I won't succeed in that. So I would also like all of you, see, there is something which I have some plans to do or I like to become, but do I really have and equipped with those skills is important. So your skills and your plans, your ambitions all should go hand in hand together. The abilities, you have to increase your abilities. So abilities, what, like towards the technology, I won't say that because you people are very much well versed with the technology. So like for us to be in the world today, we have to have these abilities to work with the technology. So whichever field you go, you have to keep on enhancing your abilities to go and adapt to the systems there. Cross-functional skills is very, very important, which we had never learned when we were in college. So like we will only stick around my microbiology class, 9 to 5, my microbiology practicals, no looking here and there and going to the college, not going to help any one of us. And at that time, we didn't have so much of internet. We were only dependent on libraries only. That was the only source for us, or our teachers who used to guide us the best, whatever knowledge they had with them, they used to really share with us. But today, you people have lots of platforms for knowledge. So please, cross-functional is apart from your subject. Please start looking more and start learning more apart from what you're doing right now. Work with diverse cultures, very, very important for that. We have to move out. Mumbai and allied regions do have diverse work people, work cultures, industries, but people down, coming from a bit of a semi-urban area, do not get exposed to the different cultures. But I would say, please go to different industries, find out the work culture. So when you're young, you can still go in different industries, explore the cultures, learn new things. So this should be also there. You should be equipped to do that. No, to understand the government policies and schemes in the sector. So please go and visit different websites, related to food. I'm not sure, like Ministry of Food Processing is one where a lot of food processing industries information is there. So please do visit, find out the different things. I don't know if I have it on my next slide, but if not, I will tell at the end of my lecture if I have more ministries where you can please visit and find out lots of figures on finance, what has been on top list of exports and agri-produce, everything. I'm going to the next, Vineet. Let's go to the next. So prepare yourself. Again, I have said, think globally. So we have to think
Hello
globally.
friends.
What is happening at the
Welcome
global
to the module
scenario?
number
What are the
4
new
of
trends?
molecular
So we have to know what's
symmetry
globally.
for
So
this
by the
degree.
time it comes to us, we know, okay, what we have to
H2
really be
half
equipped
the
for. So if you're
these
thinking
are
globally only,
different
I think vision comes to you of thinking big also. When you
images
look
of
at
each other.
the global,
This is the
your picture becomes
requirement
a bit
or
larger when we think
necessity
globally.
of
Like, see,
or
we will think
condition
of
for the
FSSAI for
measurement to
Indian
be
food
present
safety.
in a
But if I think of
molecule.
FSSC 22000,
Hello
GBR
friends,
guidelines, then
welcome to the
okay,
module
I'm
number
thinking
4
globally. So I
of
think start thinking globally, look at the bigger picture. Keep and clean and open career path. That is very important. Keep a clear. So be clear on what you want to do. Okay, you may have a plan A, B, C, D, everything is fine. But you should be clear what you want. You should be open to the career path. Open, yes, if this doesn't work, now what next? So you should be really open, and develop resilience. So I have put there what is resilience. You should have the strength, the confidence, keep motivating yourself. Protecting also from yourself, and efforts should be always there. So 100%, I won't say even 100. So efforts from each individual should be more than 100, so that any given point of time, you should not feel my efforts were less. So maybe in any age at any point in time, when since student or till date, efforts is very important, that keep putting those efforts all the time. Be ready for a change. So life has become very, very uncertain. So we have to keep changing ourselves circumstantially, everything. And then you have to have agility. Agility is just adjusting to the new things what are coming. The more you adjust, the faster you adjust, the better you equip yourself. So we'll go to and then last I have said, come out of the comfort zone. This is important because really we are very, very comfortable in wherever we are, don't want to take new challenges. So I had given an example, like I didn't wanted to learn driving a car till I was 40 because I was afraid of taking the car. I was very happy riding the two-wheeler, and yes, so I don't want to, I'm very happy, I go on my own, I'm quite independent. What's the need to learn the car? But yes, I had to push myself at that age to get out and sit in the car and learn. So there were no drivers also ready to teach me the driving. So I thought, "Nahin, madam, aise itni age mein nahi ho payega." But I convinced them, you give it a try for a few days, and I will. If you don't feel, then we leave it. So yes, I learned driving, and today I drive on the highways around 200 kilometers. So but you have to come out of your comfort zone. How much ever you find it will be difficult, but once you put in efforts, you'll get out of it. So yes, it is important. So can we go to the next? I'm just moving to the next. So when you're going for interviews, basic things, I'm not saying much because you people are really must be reading a lot on what's there in the industry. Your basics are absolutely right. They're bang on because of your curriculum, which is there with the university. But still, I would say a few things from my side. Know about the current regulations where you are going to the company. You first, any company which you go, you should know the background of the company. So on the net, read about the company before you go to answer the interviews. Try and know what are the regulatory requirements for their product which is in the market. Think of what regulations would apply to their product and the type of the industry. Common pathogens which would be there for the industry, because the pathogens in milk industry would be different, that in poultry would be different, maybe fruits and vegetables will be different. So you have to just align yourself when you're going to the interviews, be prepared. Then what are the spoilage causing organisms, testing methods, what are the different type of testing methods. This is also important, because when I was fresh and came out from the college and I was asked in one of the interviews, how do you test coliforms? And my answer was MPN. And I just could not relate it, that I can also isolate it on a plate. So there are different methods. So we, at that time, I don't know what are the books right now, but I had studied a practical book on Cruikshank, and that's the only book I remember. I don't know whether it is there now, but I said Cruikshank, and everybody, okay, smile. So I was raw from the college, so I didn't know that there are standard procedures also for even isolating E. coli. So I have put in some methods here on the slide, that there are different type of methods internationally accepted. So US FDA has a BAM, bacterial analysis manual. It's free online, you can just check. IS are Indian standards, EU are European Union standards, AOAC is American official methods for chemists. So even microbiology is part of that. There is even AFA, which is for water. So please go through this. Everything is on the net. So just go through the standard methods which are available to analyze different type of pathogens, even pesticides, aflatoxins, everything. MPN, most of the people don't know, like when basic microbiologists, we ask questions, they don't know what is the unit of MPN. So please do that. pH, I ask so many people, what is the unit? There's no unit for pH, but they keep on thinking what would be. Especially with the microbiologists, since these are the things which we don't think of too much. We know only microbiological media pH has to be 7. Okay, but what is the unit? What is acidic, basic? Please, all this is required. Temperature, pasteurizations. What is UHT? Few questions I've put because of my experience, but you can think more. Going to the next. Yeah, I have a slide on government organizations. So various government organizations which are available for food and promotion of food and standards and safety and quality, I have put here. So there's EPEDA, which is Agriculture and Processed Food Export Development Agency. Here you'll get a lot of information on what is the fastest moving food item for exports, how much it was exported, what are the regulations of importing countries for what type of a product. So one example I can give you. So if European Union is importing fish, the requirement for antibiotic residues is there. The requirement of Vibrio will be there. If we are exporting grapes or pomegranate, the requirement is for pesticide residues, Salmonella, E. coli. So it depends and changes from commodity to commodity, processed to unprocessed. So you have to think on all these lines. FSSAI, as I have told you, is Food Safety Standards Authority in India. MOFPI is Ministry of Food Processing Industries. EIC is an Export Inspection Council and Agency. This takes part like complete control of issuing the health certificates for exports and as well as in India. Then there's Ministry of Commerce, under which actually EPEDA, MOFPI, EIC, all these are coming under Ministry of Commerce because it is all concerned with the commercials. Then there is CII, which is the Confederation of Indian Industries. So it deals with industries right from mining to infrastructure to railways, you can telecom, everything. But CII has a small part called as FACE, which is Food and Agriculture Center for Excellence. Please visit the CII FACE. There are a lot of webinars now going, little courses also there on food safety and testing and quality assurance. So please, if you can go on FACE, you'll be equipped to go to the industry if you may attend these courses. So I had also said that NEPTEL, it's a consortium of IITs who have got online courses and gives also certificates, and they are all free. And they are right from aerospace to analytical chemistry to organic farming and whatnot. So if there are 2-month courses, online lectures, those are also flexible. The sports material, most of the courses are coming in July, August and September. I think July, you have to enroll in July and the exams are in September. So please visit if anyone of interested. It's such a good initiative by the government on a very big platform. So would encourage all of you to please do. I'm also doing few courses there. Not keen to give an exam, but definitely to go through those courses. Okay. So we are moving to the next. Yeah, So we are talking about opportunities. So opportunities are not going to come at our doorsteps. We have to get out and see. So I have put few slides which I'll read out. Whatever makes you uncomfortable is the biggest opportunity for growth. If I am afraid to get out of my home and go to another city, or say, okay, Delhi, I don't feel like going. I'm much comfortable. It's so unsafe. The climate is so harsh. But you have to get out from this. Make yourself uncomfortable and seek the biggest opportunities for growth. So even I went to Delhi, I was very, very resistant to go there. Didn't settle properly. But you have to get out. Then only you can see a larger picture when you go out. If opportunity doesn't knock, you have to build a door. So you usually say we have to knock a door for an opportunity, but sometimes you may not even find a door. You have to build your own door and then knock it and go inside. So opportunities follow struggles. Of course, nothing is going to come on our plate just there. So we have to take lots of efforts, huge hard work. And it doesn't come before that unless you put this hard work and effort. So this should be our constant thing, to put in lots of effort and do a lot of hard work. We should not shy away from all that. So I come to the end and I go to the next slide and say thank you for the faculty, Dr. Rajeshri, Shubhida. I know both of you, but not much there. But thanks a lot. And thanks audience for the patient listening. Maybe we can go through few questions. I'll be on mute and maybe we can take few questions from your side.
There is a question, Sandesh. Hello, Sandesh. Hello, Sandesh.
Yes.
There's a question.
Yes.
Sandesh, you have some questions coming.
There are no questions.
Sandesh, you mute yourself. I will take this. There's some issue with your audio, I guess. Yeah. Sonia Madam, it was really wonderful presentation, and I'm sure our students have learned a lot. And thanks for accepting our invitation and coming here. I see one question here. Hello.
Yeah, I can hear you. Please go ahead.
Okay. Akanksha is asking, biotechnologists can also go for food industry.
Yes, Akanksha, definitely you can go in. As I said, you have to do cross-learning a lot. Since you are from life science background, you have the basic knowledge. Go and do a lot of cross-learnings. It is open for you. The food industry is open, no problem.
Right. Any more questions, students, teachers? I think you have given such wonderful information encompassing all the possibilities and how to prepare. I think that's why they don't have any questions.
Good.
And over to Darshana, who's going to take it?
Yeah, ma'am. Mrudal ma'am is going to go ahead.
Yes.
There's one more question. There is one more question from Vinith.
Okay. Yeah.
Can you take the question?
Yeah. Okay. There's a question from Vinith. There is a growing trend of eatable cutleries. What is your take?
Yes, there is, and it is, if tested, yes, but what I still feel, the eatable cutleries are there because they are biodegradable, as well as they're made from plant, as well as they're made from, I have come across few cutleries which are made from pulses. And yes, we can digest pulses also. And since ages, the gram flour is one of the which has a lot of properties of even coming together a lot. So you have a lot of cutleries coming, and yes, they are good enough, but the testing has to be there. I insist, if you buy any of these, please ask for certificates.
Yes. I think over to Mrunal.
Yes. Hello, am I audible
Yes.
to everyone?
Yes.
Okay. Good afternoon, all. Our distinguished speakers of the webinar, staff members, and students. I am Mrinalini Patil on behalf of Microbiology Department of KARMAVEER BHAURAO Patil College. Extend a very hearty vote of thanks to the honorable delegates for gracing today's webinar and giving their valuable time. I would like to express my sincere gratitude to Dr. Arun Karaj sir for making this webinar meaningful. It's really interesting to know that microbes are 10 times more in number compared to body cells, and bacteria absorb anti-cancerous compound. Thank you, sir, for provoking such thought that we have to maintain our natural microbial flora. So it's like in single dish, you have offered variety of flavors in a form of information for utility of microorganism. Thank you, sir. I extend my heartful gratefulness to Mrs. Sonia Nandre, madam. The way you explained, madam, the subject was extraordinary in spite of having technical issues. Your enthusiasm led us in understanding of various opportunities in the food industry, in various sectors of food industry. Our student will definitely benefited by knowing the opportunities as well as the certification you guided and various government agencies. So it's a good coverage to the subject content. Thank you so much, ma'am. I once again thank you, Dr. Arun sir and Dr. Sonia madam, that bringing our attention to cross functioning and guiding for interview preparation also. I would like to express our profound gratitude to our principal, Dr. V.S. Shivankar sir, for giving opportunity to arranging such events. I would like to thank our beloved head of department, Dr. Shubhada Nayak madam, our
And I
guiding
had to,
talk, source of
according to
inspiration.
this, why
I
to
must
waste
mention
what then now present
big
I think the
thank
job
you to Dr.
rotation
Rajeshri Ghorpade madam for
H5
taking
in this
tremendous
to the
efforts in
next
arranging such
rotational it
webinar.
is methane
I
molecule
must
like this.
mention deep sense of
We have
appreciation
tried
to Mr. Vinit
times we
Kala,
have rotated
Ms. Darshana Raut, Sarang sir, and I'm happy to express my vote of thanks to our staff members who made this webinar into grand success. Finally, the wonderful student, you have turned up in such a great numbers, and thank you for your patiently listening. Thank you once again all for your cordial cooperation. Thank you very much.
Thank you, Mrinal. Thank you all. Thank you for joining. Thank you so much.
Thank you, ma'am.
Thank you, Dr. Kharad. Thank you, Sonia madam.